Here are our learning objectives for Chapter 17, and nothing to kick off a chapter like a warning. As ever, there are many similar moving parts between the processes I described in the previous chapter, DNA replication, and the processes that I am about to describe, RNA transcription, and polypeptide translation. These are all recipes for polymers, so they all have a beginning, a middle, and an end. Initiation, elongation, and termination. This can lead to confusion, but you need to carefully manage these processes in different bins in your mind. Again, I will ask questions about this, and it will be confusing if you don't keep the processes separate from the beginning. We know that the sequence of nitrogenous bases in the nucleotides of DNA are where all the information is stored. Somehow, those sequences lead to a collection of cells that become unique individuals of every species. The link between DNA and all this is proteins. So in this chapter, we'll explore how DNA produces different proteins through the process of gene expression, also known as transcription and translation. Before it was even widely accepted that DNA was the stuff of inheritance, the link between genes and proteins was found through experiments with a fungus called Neurospora crassa, otherwise known as red bread mold. Similar to Thomas Hunt Morgan, George Beadle and Edward Tatum generated mutants of their model organism, in this case using ionizing X-rays to change the DNA. They grew their fungi on agar media, and the wild-type fungus was able to grow on a minimal medium that included very few nutrients added to it. They produced mutants that could grow on richer media that had additional food sources, but not the minimal media. By crossing the different mutants, they were able to identify 3 different types of mutants that were unable to synthesize the amino acid arginine. It was known that the wild-type Neurospora that could grow on minimal medium produced arginine using a 3-step metabolic pathway. They reasoned that the 3 classes of arginine mutants were each deficient in an enzyme necessary to produce the amino acid. This led them to their one gene, one enzyme hypothesis, which states that each gene dictates production of a specific enzyme. Let's take another look at Beadle and Tatum's experiments. The wild-type Neurospora crassa was able to grow on minimal medium and able to synthesize arginine with no other chemical additions. Their mutagenesis produced mutants that could grow on media supplemented with arginine, but unable to grow on the minimal medium. Further experiments showed that their arginine-deficient mutants were not all of one type, but could grow with addition of different supplements to the minimal medium. They concluded that there were 3 different types or classes of arginine mutants, each one able to survive with the addition of a supplement that was an intermediate in the arginine biosynthesis pathway, ornithine, citrulline, or arginine. Their 3 mutants were each lacking in an enzyme from the pathway. One more time. Beadle and Tatum used X-rays on the fungus Neurospora crassa to generate 3 classes of arginine mutants. Their experiment, published in 1941, proposed the one gene, one enzyme hypothesis, though now we realize that proteins do a lot more than just function as enzymes, so now that hypothesis has been expanded to the one gene, one polypeptide to reflect on this fact. For their discovery, they were awarded the Nobel Prize in Physiology in 1958. How does DNA make polypeptides? The bridge is RNA. DNA is a molecule that cells would rather not have manipulated any more than is strictly necessary. RNA is a molecule that is a lot more tolerant of manipulation. DNA is like a pristine library of information, and RNA is like a photocopy that you can take notes on, mark up, and not feel too badly about losing or damaging. The first step in making a polypeptide is to make an RNA version of the DNA molecule in a process called transcription. The RNA copy is called a messenger RNA, or just an mRNA, now that we are familiar. The mRNA is then translated into a polypeptide at the ribosomes. The process is similar in eukaryotes and prokaryotes, in some ways, with a big important difference. That difference is the nuclear envelope. In bacterial cells, for example, transcription and translation occur simultaneously since there's no physical barrier between DNA and ribosomes. In eukaryotes, not only is there a barrier between DNA and ribosomes, but the barrier makes it possible, no, necessary, for the naive mRNA to be processed and modified into a mature mRNA before export to the ribosomes. This brings us back to a topic we've seen before, I'm pretty sure. It's called the central dogma. It's been called the central dogma of biology, of molecular biology, of genetics, but the important wink thing is this. DNA encodes RNA, which encodes the polypeptides. In this order, in all living cells, almost always, and I'm not going to tell you about reverse transcriptase, so please don't ask. Remember that DNA was suspected as the molecule for inheritance, not certain at first, because it wasn't known how 4 nucleotides might encode 20 different amino acids. Forget that Samuel Morse had managed to create a code for the 26-letter alphabet and numbers and punctuation marks using only dots and dashes, but there it was. DNA was the molecule encoding instructions for proteins, but how? The answer was found in the genetic code. How many nucleotides are necessary to code for 20 different amino acids? One nucleotide with 4 different options, A, T, G, or C, could only encode 4 different choices. 2 nucleotides in series, each with 4 different options, could encode 4 times 4 different options, or 16 different things. That would be like AA, AC, AG, AT, all the way to TG and TT. 16 choices. 3 nucleotides following this pattern would produce 4 cubed, or 64 different options. 64 is more than 20, and that is plenty for encoding amino acids, everything from AAA to TTT. So 2 nucleotides can't quite encode 20 different amino acids, but 3 nucleotides definitely can. We call these sets of 3 nucleotides codons. Nucleic acids can relate the amino acid sequence by using a triplet code. Every 3-letter series of nucleotides in the DNA or in the complementary RNA means a specific amino acid. So a more informative version of the central dogma looks like this. DNA provides a template for the sequence of nucleotides in the messenger RNA. At the ribosomes, the triplet code can be decoded as a series of amino acids. Transcription comes first. One strand of DNA, the template strand, is used to make an RNA copy. For a particular gene, the template is always on the same DNA strand. In translation second, the messenger RNA codons are read in the ribosome in the 5' to 3' direction. Each codon dictates the amino acid to be added to the polypeptide in order. Given that there are 64 codon options, 61 of them encode the 20 amino acids. The last 3 are called stop codons because they stop the translation process. This means that there is some redundancy in the genetic code. Each amino acid has, on average, over 3 different possible codons. But no codon can code for more than 1 amino acid. There is no ambiguity. And since each codon has a meaning, the codons have to be read in the correct reading frame or the proper order of triplets for the correct polypeptide to form. Since I haven't formally introduced you to this table, let me do so at this time. This is the codon usage table showing exactly how the RNA triplets encode the amino acids. Please do not attempt to memorize this. You should, however, learn how to read the table, though. Note, on the left edge here, over here, are the first RNA nucleotides in the codons, starting with U, C, A, or G. The top 4 boxes all start with U, and the bottom 4 all start with G. The second mRNA base, indicated along the top of the table, follows a similar pattern for the second position in the codon, U, C, A, or G. And for the third RNA nucleotide, we look at the right-hand side of the table, UCAG, UCAG, UCAG. So, for example, if we wanted to know what the triplet AUC encodes, we look on the left edge for A. There it is. Then along the top for U, and then over on the right edge to C, and we could find AUC, which encodes for the amino acid ILE, which is biochemistry shorthand for isoleucine. Notice that there are 2 other codons that encode isoleucine, AUU and AUA. That is the redundancy feature. More than one codon can encode the same amino acid. But those codons can only encode that one amino acid. They can't sneaky encode some other amino acid, or there would be chaos. That is the no ambiguity piece of the puzzle. Often, the first 2 nucleotides are the most important, and the third one has to be there, but it doesn't change the amino acid encoded. GCU, GCC, GCA, and GCG all encode alanine. GC is the most important part. The third nucleotide is less important, though something has to be there in that third position, or again, the system falls apart. I did say do not memorize the whole table. I only want you to memorize 4 codons: AUG, UAA, UAG, and UGA. AUG, which you may have caught a glimpse of being right next to AUC, and the rest of the isoleucine codons, is the only codon with more than one meaning. It encodes the amino acid methionine, but it also encodes start. This tells the ribosomes where to start translating the messenger RNA. Oddly, but not too importantly for us, is that AUG is the only codon for the amino acid methionine also. UAA, UAG, and UGA are the stop codons. These do not encode any amino acid. They signal the ribosome that it's time to wrap up this polypeptide and let other processes take over. Being at Auburn, maybe you can remember that the ones we need to stop are UGA, like University of Georgia, UAA, as in University of Alabama, and UAG, as in University Alabama is gross. Codons are always 3 letters that can be read at the ribosomes, and the 3-letter words can't overlap. If any letters get added or subtracted, the message changes, and usually the change is from sense into garbage. For example, the first sentence of 3-letter words is an odd but still sensible sentence. The cat ate rat and eel. If we remove one letter, the R in rat, for example, and try to read the sentence as a series of 3-letter words, it becomes nonsense. The cat ate ata, nde. Or if we remove one letter at the beginning, all sense is lost. The sentence is fatally flawed with no sensible words. All I did was remove the first letter and everything falls apart. Hek ata, ter ata. This can also happen with messenger RNAs, although, as I just showed you, all 3-letter combinations have meaning, but the resulting code may give you a garbage protein.